Hello, everybody, and welcome. On behalf of PAEA, AAPA, the PA Foundation, and the National Institutes of Health, I'd like to warmly welcome you to today's webinar. I'm Dr. Nina Multak, the Associate Dean and Director of the University of Florida School of PA Studies. Thank you so much for joining us today for an exciting presentation and conversation about research, genetics and genomics, precision medicine, and the NIH All of Us program. First, a few housekeeping items. I'd like to remind you that we're dedicated to providing a harassment-free experience for everyone, and we appreciate the respect and courtesy you extend to one another in these virtual spaces. If you have any questions during the sessions, we'll be glad to respond to as many as possible. Please place your questions in the Q&A area at the bottom of your screen. And we'd like to ask you to use the chat for any technical or other issues that you may encounter during the webinar this morning. Today, I have the pleasure of introducing our guest speakers and colleagues, and I'll provide a brief biography prior to each of their presentations. Here you'll see several key definitions that you'll want to familiarize yourself with prior to the initiation of our presentation today: engagement, outreach, recruitment, and retention. If you'd like to just take a few seconds to review these definitions, that'll help you understand best the content that'll be presented this morning. Next slide, please. Gives me great pleasure to highlight each of our speakers this morning. Dr. Sheri Schully is from the National Institute of Health All of Us program. Dr. Nicholas Hudak is from Duke University. He'll present on research. Dr. Nguyen Park is the president of the Society of Genetics and Genomics. And Dr. DeShana Collett is from the University of Kentucky, a professor, and will highlight her talk with a focus on precision medicine. Here's a good look at our agenda this morning. We'll have two presentations followed by a brief break, the second two presentations, and we'll wind up with your question and answers. So here we go.
Hi, everyone. Sorry, go ahead, Nina. Sorry.
I'm going to introduce you and we'll let you take it away. Dr. Sheri Schully, PhD, is the Deputy Chief Medical and Scientific Officer of the All of Us Research Program. Prior to this role, she was a team lead and senior advisor for disease prevention in the Office of Disease Prevention. There, she led the effort to systematically monitor NIH investments in prevention research and assess the progress of that research. She also served as the team lead for the knowledge integration team, as well as a program officer in the epidemiology and genomics research program at the National Cancer Institute. She came to NCI as a presidential management fellow in 2005. Dr. Schully has authored and co-authored more than 70 papers, many focusing on the translation of genomic technologies into clinical and public health practice. She earned both a PhD in biological sciences with a concentration in population genetics and a BS in zoology with a minor in chemistry from Louisiana State University. Sheri, please take it away. The stage is yours.
Thanks, Nina, and sorry for jumping the gun there. I'm just so excited to talk to this group that I wanted to go ahead and get started. As Nina mentioned, I am with the All of Us Research Program at the National Institutes of Health, and I'm really excited to be here today to really talk about not only our program, but then we can hear from the other three speakers today and really how you as PA students can really leverage this program to look at research, genetics, and precision health. Next slide, please. So the All of Us Research Program's mission is to accelerate health research and medical breakthroughs by enabling individualized prevention, treatment, and care for all of us. And we're doing that in three ways. The first is we're nurturing partnerships with 1 million or more participants for decades. And those participants really reflect the broad diversity of the United States. With that engagement with our participants and their data donation to the program, we hope to deliver one of the largest, richest biomedical data sets that is both broadly available and secure. But just because we have this unique engagement with our participants and this great infrastructure doesn't mean that this is going to be, if you build it, they will come. So we really need to catalyze an ecosystem of communities, researchers, and funders, including folks like you, who make all of us really an indispensable part of health research throughout the time that it's around. Next slide, please. So at All of Us, we really want to understand the whole person. And so we are really collecting data from individuals that really get at not only their biological and clinical issues that might be going on, but also what their environment looks like. What does their social environment look like? What does their behavioral aspects look like? And all of these together really help to help us understand what diseases and or wellness factors certain individuals may have, and really help us focus on really individualizing the treatment, care, and prevention for that individual. Next slide, please. At All of Us, we have quite an extensive protocol, which I'll go through each step of this, but overall it includes usage and authorization to use electronic health records in research. Our participants answer surveys over time about various topics. They provide physical measurements as well as biospecimens to the program, and they can even share data from wearables, including their Fitbits and Apple Health Kit. Next slide. Next slide, please. Currently in the program, we have 615,000 participants who have signed up to be in the program and have actually gone through a consent process. Of those, 428,000 have actually completed all the initial steps of the program, meaning that they have consented to let us use electronic health records in research, have answered our first 3 basic surveys, and have contributed a biospecimen to the All of Us research program. On the right-hand side, you can see sort of our enrollment over time. We really did have a really good clip going until COVID hit in March 2020. I think you're going to hear this from many researchers as people talk about sort of what they had to do during COVID. We couldn't have in-person visits. That wasn't the best thing to do for our staff or our participants. And so we really had to pivot and do research in a different way and really find out how to meet people where they are. Since we couldn't necessarily bring folks in for an in-person visit. And so we are climbing back up now, trying to get to that 1 million person cohort that we're having. But we did have a little bit of a setback, but we're on track to get there. Next slide. All of Us really has a commitment to diversity of our participants. We have participants from all 50 states. And you can see here on the left that the map of the United States that really shows our geographic diversity. The darker states indicate where we have more participants actually within those states. And we have efforts going forward to sort of fill that middle part of the country that's a lighter shade of blue, because we want to really make sure that we have the geographic diversity to not only bring in folks who are in more sort of urban areas and close to regional medical centers, but also those in rural populations. We have a commitment to diversity, not only for race and ethnicity, but you can see that graph here on the right, where we want to have 50% or more of our participants be diverse in racial and ethnic minorities. But we also want to make sure that we're also looking at folks who have been historically underrepresented in biomedical research. And so for us, that definition includes not only racial and ethnic minorities, but as well as sexual and gender minorities, people who come from low income groups, low education, and other groups that really have not been part of research to date. Next slide, please. I mentioned already some of the data types that we currently have available. So here are some of the examples of how much data we currently have on our participants. So we have As I mentioned, surveys from over 372,000 participants that have actually done surveys and answered for themselves self-report data on what their experiences have been for several different surveys, including basic demographics, personal and family health history, and many others. Over 306,000 participants who have done those physical measurements in person. So those include blood pressure, heart rate, BMI, and more. We have actually obtained electronic health records from 258,000 participants. And so we have actually been able to make that connection and bring the data in for over 258,000 participants. With those biospecimens that our participants graciously donate to the program, we have actually been doing both whole genome sequencing as well as genotyping arrays on everyone. And so currently we have almost 100,000 whole genome sequences and over 165,000 genotyping arrays on our participants, and almost 13,000 individuals who have donated data on their wearables, so we can really get a sense of what their movement is like throughout the day. We will eventually be bringing in sleep data from some of these activity monitors and things like that. And then we also store a good set of biospecimens that we will be using in forthcoming assays and for allowing researchers to use for their own purposes. Next slide, please. So I want to give you a little background on sort of how you might go about getting to this data, this really rich data source that we've created. Certainly, PAs can also be involved in helping get some of our, enroll some of our participants and help with that engagement piece. And we definitely have resources about that. But I'm speaking here more from the how you might use the data to do a research project yourself, or for some of your students that may be wondering how they might be able to get to this really rich data. So our data access has 3 tiers. The first one is a public tier, and I'll walk through some examples of that today, but these are open to everyone. There's no login required. You can actually log in and see some of this in action at the very high aggregate level. And so I'll go through, as I mentioned, some of the examples of this, but it really gives people an idea of what's in our database before they decide if they want to actually apply for access to the more, for the higher tiers, including the registered and controlled tiers, which are really requiring access, but that's sort of where our genomic data lives. Some of our more individualized data that we just can't let that out to the public, that public tier really needs to be aggregate level data only so that people can't be identified. But those registered and controlled tiers have a little bit more granularity that helps you really do some in-depth research analyses. Next slide. Here's an example of sort of the data and how we've put it out over time. And so here we are in 2023. We have a big announcement coming out, including some new data that's going to be released tomorrow. So be on the lookout for that. So some of the numbers you've seen earlier in my presentation will be changing greatly because we have so much more data that we've ingested into the ecosystem. And so I've already mentioned several of the things that we have ongoing now, but over time, this resource will grow. And not only because we're collecting more participants and growing sort of the number of participants that are in the system, but also because we're bringing in new data types. And so our participants will continue to do additional surveys over time for areas that really matter for the program. We will be integrating different wearable data, including sleep, as I mentioned before. We will have linkages to external data, including census and some of the social vulnerability index, the environmental justice index, and things like that to really enrich the data that we already have. And we will be doing additional assays and things like that. So beyond just whole genome sequencing and genotyping arrays, we'll be bringing in other data on the specimens that we've collected and stored. Next slide, please. So in order to become a researcher and get to that sort of registered and controlled tier of the data, it's actually fairly straightforward. I put a QR code here just so that it's easy for folks to get to, but it's at researchallofus.org. You can really use that public site to look at what data is currently available within the cohort. Then you check for your institutional agreement. And so many institutions across the US, over almost 400 institutions have what we call institutional agreements with the All of Us Researcher Workbench. If your institution already has an agreement, then you move on to step 3. If not, it's pretty easy to set one up and we can help you out with that. You would register as a researcher, go through a identity verification, and then take a responsible conduct of research training, sign a data user code of conduct, and then you're actually into the data, which we've had reports of people who, if their institution already has an agreement, going from step 3 to step 6 in under 2 hours and having access to really incredibly rich data, which is really unheard of until now. Next slide, please. If you're not ready to jump into the data just yet, but you want to keep up with the program and see what's going on, not only from like an enrollment side and what's happening with our participants, but also what's happening with the research and with the data that we've put out there for folks to use, you can sign up for our Researcher Roundup. This is sort of a bi-monthly newsletter that goes out that really highlights the exciting things happening with All of Us. It's a good way to keep up with the program and just keep in mind additional things that might be coming, including additional data types that are coming on board that may be of interest to you. Next slide, please. So certainly us at the All of Us Research Program within NIH are not doing this alone. We have an incredible community and provider partner network who's helping us not only recruit and enroll these really diverse participants, but keep them engaged with the program over time. This is a longitudinal study, so we want to be following folks over time, seeing where things change, and really being able to track disease states over time. And that's multiple disease states. We're not focused on any one disease, and we can't do it alone. Next slide. And then certainly our consortium members, which are the actual sites that are bringing folks in, not only within the community, but also within healthcare provider organizations, some of our genomics partners, as well as the folks who help us design all of our technologies to interact with our participants, as well as make this data available to the wide community. Next slide. So with that, I want to thank you and turn it back over to Nina.
Thank you so much, Sheri. I do want to share with you and with all those listening today that I did sign up. I registered to be a researcher in All of Us, and I also signed up to receive the newsletter. So I encourage everybody listening today to do the same. Thank you so much, Sheri.
Perfect.
We'll hold the questions until the end. With that, we'll move on to our research focus, and our speaker today for our research focus is Dr. Nicholas Hudak. Dr. Hudak has been a clinically practicing PA in neurology since 2004 and a PA educator since 2009. He is currently an associate professor in the Duke University School of Medicine, Department of Family Medicine and Community Health. He serves as a clinical coordinator in the Division of PA Studies and as an assistant director in the Duke Center for Interprofessional Education and Care. Dr. Hudak has earned master's degrees in PA Studies and Education and Counseling, as well as a PhD in Educational Leadership, Policy, and Human Development from North Carolina State University. Dr. Hudak has authored or co-authored 18 peer-reviewed articles across several publications, and several research-related achievements in PA education include the AAPA PAEA Research Fellowship, PAEA STAR Program, the PAEA Peterson Grant recipient. He is currently an assistant editor of the Journal of PA Education. We'd like to warmly welcome Dr. Nick Hudak for Talk on Research.
Thank you so much, Dr. Multak. I appreciate the generous introduction, and I am excited to be here today. Next slide. So briefly, no disclosures. And the next slide will list our objectives for this session. So as Dr. Schully shared, One of the major goals of the NIH and the All of Us program is to accelerate research. Research is an immense, complex topic. So in the next 25 minutes or so, I'm going to present a high-level overview of research to help all of us have a shared understanding of research principles and processes, as well as help all of us think like researchers and for you to consider how you can expand your research network. This session will also serve to set the stage for the more focused presentations on genetics and precision medicine. As healthcare professionals, health professions educators, and health professions students, it is imperative that we appreciate the high level of knowledge, skills, and attributes necessary to conduct high quality research. So I hope that sentiment will be a takeaway for you in this session. Next slide. So as we begin this presentation on research, it's important for us to have a shared definition of what research is. The Department of Health and Human Services defines research as a systematic investigation, including research development, testing and evaluation, designed to develop or contribute to generalizable knowledge. In short, research is about extending current knowledge and the discovery of new knowledge. As Dr. Schully shared, these are medical breakthroughs that we hope to bring to our patients and communities. The design and systematic investigation in research can have different purposes, different samples, different populations, as well as employ various methods. So in this presentation, we're going to take a close high-level look at 5 types of research listed on this slide. Before doing so, it's important to recognize that research is a regulated practice, given the potential impact and risks that come with new knowledge generation. Next slide. One primary goal when conducting research is to protect the safety of the research subjects, whether animals, humans, or populations. Safety should also be considered with regard to the integrity of funding, the research processes, as well as the data that result from research. In clinical research, which is what we think a lot about, the privacy of patients and the safety of human subjects is of utmost importance. Other key regulatory factors include conflicts of interest among those involved in research, the integrity of data and how it is shared, as well as consideration of the regulatory burden upon those conducting the research. New knowledge is derived from research data, and all research data is susceptible to both error and misinterpretation. So data integrity refers to ensuring that the data are complete, verified, undistorted, and free from bias. There are various procedures used to ensure the integrity of data, but they can vary greatly from field to field. They may include review of data within research groups, replication of previous observations and experiments, peer review, the sharing of data and research results, and the retention of raw data for possible future use. Next slide. So Dr. Schully spoke to asking questions about the All of Us data. So to meet the objectives of this presentation and guide our overview, I'm going to propose 5 questions every current and prospective researcher must ask themselves. What is the research question? What type of research do I need to conduct to answer that question? What research design and methods are necessary? What knowledge and skills are needed to conduct the study? And then a bit separately, how can I develop a PA-focused research network? So we're going to answer these questions throughout our presentation. Next slide. The first big question is, what is the research question? So to answer this question, we need to consider how to approach answering the question and what methods are needed to answer it. The foundation for research design is based on inquiry or theoretical frameworks, as well as conceptual models. The selection of a framework and the development of a conceptual model are informed by the current literature. We cannot extend the current literature if we don't know it. Conceptual frameworks and inquiry frameworks provide a lens through which the investigator approaches the design of the study, as well as how the findings are interpreted. Research design is also informed by what types of methods are most appropriate to answer the research question. What tools are needed? Research methodological design may be quantitative, qualitative, or involve both quantitative and qualitative methods, which is known as mixed methods. Quantitative studies may involve analysis of primary data conducted during the study, secondary data available from other sources, or meta-analyses, which is an examination of data from multiple independent studies on the same subject to identify trends. Qualitative studies may include interviews, focus groups, observational methods, ethnographies, which look at descriptions of people and groups, as well as document analysis. And then some studies may be mixed methods involving both quantitative and qualitative, or there may be multiple method studies, which include multiple quantitative or multiple qualitative methods. So giving our shared understanding of the definition of research, the regulatory ethical consideration, and knowing about inquiry frameworks, we will proceed to the other big questions. Next slide. Once a research question is known, the investigator moves on to answering what type of research am I conducting and what methods do I need. So right now we're going to sort of look at the question of what type of research am I doing? Is it basic science? Is it clinical? Is it improvement science, health services and workforce, or is it educational research? For each type of research listed on this slide, I will briefly describe some of the more common investigator disciplines, the principal areas of focus for samples, units of analysis, and common types of studies. This general overview will also function to provide a context to classify genetics research and better understand precision medicine in the upcoming presentations. It's important to remember that this is a brief overview, so it's neither comprehensive nor exhaustive. And it's important to recognize that as a PA, who can be investigators and co-investigators, you may be involved in clinical research, improvement science, health services, and educational research. So you may think, oh, PAs aren't involved with this, but we are not only involved with it in terms of practitioners of evidence-based care, but we can also be contributing to research in these areas. Next slide. So first, we're going to take a look at basic science, fundamental, or bench researchers, multiple terms to describe one discipline. So bench research provides foundational knowledge for the applied sciences, including clinical research. Investigator disciplines are listed, and the focus here can be at the atomic level, molecular level, cellular level, as well as genetic level, which has implications for the data sets you've learned about in the All of Us research program. Precision medicine, which you'll learn more about, does involve genomic sequencing to identify biomarkers, which can be used to better understand health and disease. These can be used for prognostic purposes as well. So, for example, in primary care, precision medicine could be used to inform decisions regarding treatments such as smoking cessation by examining a patient's speed of nicotine metabolism. In oncology, precision medicine can inform selection of the most effective treatments and avoid ineffective treatments with harmful side effects. Dr. Schully shared that data collection includes biospecimens such as blood, urine, as well as biometric data. So a lot of this data from the basic sciences, I provided examples of how it can be applied in clinical settings. So don't think that this is far afield. It's really providing a foundation. Next slide. So clinical research is one we think a lot about as healthcare practitioners who go to the literature to inform our practices. And of course, clinical research draws from many disciplines, including epidemiology, biostatistics, and health decision sciences, to name a few. These studies can be experimental and interventional. This is where we think about our phase 1 and 2, as well as 3 and 4 studies, which can be focused on safety and then subsequently efficacy and generalizability of findings. Studies can also be observational, looking at groups of or populations, looking at cross sections of populations, as well as looking at case control examples. So we read a lot of clinical research as healthcare providers. And again, this is looking at sort of the impact on human subjects, which is the patients that we care for. Next slide. So improvement science, also known as implementation and knowledge translation sciences, really look at how we can facilitate the uptake of clinical research into practice. How are we applying it? Is it being applied, yes or no? How well is it being applied? What impact is it having? So to answer those types of questions, we of course look at multiple disciplines, which look like some of the disciplines from before, but we also look at psychology, sociology, and economics. And again, the unit analysis here is going to be not just patients, but really what are the providers doing? What is the impact of policy? What's the effect on populations? Next slide. Next, we look at health services or workforce research, which draws from similar disciplines. And the focus here is precisely on health systems delivery improvement, looking at practitioner levels, but also health systems as a whole, as well as the role of payers, policy, and of course, the ultimate impact on patients and outcomes. And there are various types of studies that are used in this type of research. Next slide. Finally, we have education research, which I think very much falls into the social sciences realm. So you'll see disciplines from education, psychology, sociology, as well as statistics. And the focus here is on workforce development, not we think about the pre-licensure training, which is the learners who are enrolled in training programs, but we also think about the continuing education of that workforce. We look at the retention of the workforce and what types of programming and incentives can develop the healthcare workforce. And there can be various types of studies, again, that look at effective methods for education. And again, for both pre-licensure, but also continuing education. Next slide. Now that we've reviewed the 5 major categories of research, next, an investigator should be able to determine what type of research they should be conducting to answer their research question, as well as what methods should be employed. So next, we'll move on to answering the big question, what knowledge and skills are needed to conduct this study? This is a question about us as individuals and any potential collaborators that may be involved in research with us. So to answer this question, we need to be familiar with the steps in the research process: guiding principles, rules, specific skills, data sources, as well as the process for disseminating findings. And you received a preview of this in Dr. Schully's overview of what steps need to be taken to be a researcher and access data from the All of Us program. So at this stage in the process, investigators need to be mindful of ethics and safety and inclusivity, as including appropriate engagement with the institutional review board. Next, investigators will collect primary data or obtain available secondary data, then analyze the data with appropriate systematic methods. Once the study findings are identified and interpreted, they need to be contextualized with reference to the extant literature, and then the investigators are responsible for describing the findings, if and why they are meaningful, what these findings mean to current practice, as well as future research. This is often described as the what, so what, and now what of research. Finally, the authors may consider dissemination of findings through presentations and scholarly publications. Next slide. Throughout all the steps in the research process described here, guiding principles and rules should remain at the top of mind of the investigators. Researchers are expected to demonstrate high levels of ethics and integrity, as well as be rule-abiding in terms of maintaining required trainings, interaction, and approval from their institutional review board, and following through on other agreements. So we'll go to the next slide. So this slide is going to provide more details about these guiding principles and rules. So we need to be cognizant of factors related to justice, equity, diversity, inclusion at all stages of the research process, including when determining the research question, how the study is designed, how data is gathered, and how findings are interpreted. Dr. Schully described how the All of Us research program has set goals to have representation from diverse communities and engage with communities in their research efforts. And I think this is an exemplar. Furthermore, researchers should involve relevant stakeholders within the research design process, as well as appropriate collaborators who bring necessary skills to the study team. Researchers also need to ensure results are disseminated, whether or not the findings are significant or non-significant. Next slide. This slide lists several specific research skills investigators should have. Researchers should have a high level of intellectual curiosity while being able to hold an objective stance for the research questions they are answering. Researchers also need sufficient organization and time management skills for research planning and implementation. As described in the research steps a few slides ago, researchers have to have a high level of skills to implement systematic study methods, whether quantitative, qualitative, or mixed methods. Investigators must also have a high level of critical thinking skills to review the literature, to develop the research question, to interpret, disseminate the findings. And as before, researchers must understand principles of research as well as the regulatory rules related to research. Next slide. Of course, data is necessary to conduct any type of research. So as shared before, data may be considered primary or secondary based on whether it is collected during the study or is available from other sources. This slide lists data sources by type of research. And I would like to specifically share potential data sources in the health services and education categories, including from national PA organizations. Dr. Schully described how the All of Us program is involved in data collection and analysis, and the great amount of data that is available, as well as the need for data to be accessed with proper training and through data use agreements. And again, there are hundreds of thousands of data points available from the All of Us program, from surveys, physical measurements, electronic health record, genomics, and wearables. Next slide. So upon completion of a research study, once a researcher has gone through all those steps, the investigators may disseminate their findings in different formats, including poster presentations, published abstracts, podium presentations at conferences, as well as manuscript publications in journals. The peer review process is one means by which quality assurance is better ensured at the time of dissemination. Peer review is customarily involving the review of your scholarly work or the researcher's scholarly work by at least a few peers who are knowledgeable about the topic area. Peer review manuscripts also should involve journal editors. There are a variety of publications for investigators to consider based on their area of research. In the PA profession, some health professions, or I should say health services research is published in JAPA, and research on pre-licensure and post-licensure education is published in the Journal of PA Education. Precision medicine journals include the Journal of Precision Medicine, the Journal of Personalized Medicine, and the Journal of Laboratory and Precision Medicine. These are just a few, and I wager there are going to be more and more over time as this area and field evolves. So while dissemination of study findings is considered the last step in that list of the research process, it's important to recognize that it does set the stage and inform the designs and needs for future research, in addition to impacting our current understanding. Next slide. So now we'll move on to our final big question. How can I, whether a clinically practicing PA, a health professions educator, or a PA student, how can I develop a PA-focused research network? We will answer this question by conducting an environmental scan of available resources, explaining the networking process, identifying potential sources of funding, and imagining your first or next research study by returning to that big question of what do I want to study? Next slide. So a researcher should never feel alone in this 21st century where more than ever before, we have a high level of access to people, the literature, computing power, and continuing education. Human resources can be some of our greatest resources, including peers who are involved in research, potential collaborators, mentors, librarians, statisticians. So at any stage of the game, and also recognize that even your peer reviewers and the editors of journals and reviewers for conference materials are on your side in terms of enhancing quality. But just as I sort of underscore the importance of human resources, I actually list the literature first, because the extant literature can be one of your greatest resources. Because as before, you want to be knowledgeable about current literature before you can extend it. It's also important to know, going back down and thinking about computer software, that of course we are at a time where we have lots of access to different types of technologies with quantitative and qualitative methods. And of course, machine learning and artificial intelligence is, I think, increasingly in our view as tools that researchers can harness. And finally, you may recognize the need for your own continuing education to develop skills necessary to conduct research. This can be done through non-degree programs as well as degree programs. Next slide. So networking is likely a skill you already possess, but it's one that I believe we can all always be working on. So connecting with others can occur in in-person and virtual settings, as well as through individuals that may be connectors to other people, or individuals you know who are willing to make introductions. So some single communications is a great way to start, but to continue your networking, you may need to be following up with people. And this may be asking for help in terms of your own work. It also may be offering help or seeking mentorship. Investigators should network with individuals within their own profession, but also individuals beyond their profession. The quality of research design and implementation is more often than not strengthened by interdisciplinary and interprofessional teams. So you can also grow your network through online tools such as social network and particularly professional social networking sites. And attending events like this can also help you identify people you may want in your network. Next slide. So given the time and skills necessary to develop and implement a research study, investigators may seek funding to cover the costs of time and other research expenses, such as computer software, consultation with statisticians, as well as the costs may be associated with obtaining data. Funding may come from within your organization or outside your organization. Requests for funding typically involve a detailed application process, as well as requirements for reporting and accountability. Generally, investigators may begin earlier in their career by pursuing small grants before pursuing large grants. And newer investigators can also benefit from collaborating with more experienced researchers who have a record of being awarded funding. Lack of funding can be considered a barrier to pursuing research. So I would just encourage you to see it as a barrier, but not a disqualifier, and that you may be able to be involved with research without funding. And as you consider potential research in your future, keep in mind, again, that you may not need funding or large amounts, but this is something you'll have to consider with respect to your research design. Next slide. So I want to conclude with questions for you to think about in terms of your career and research possibilities. Think of maybe a topic or two topics that interest you as a provider, as an educator, as a learner. Consider topics that are related to what you're already working on or that the impacts you want to make. With a topic in mind, you should ask yourself what you know from the literature, what you might need to look up and learn more about, what you need to understand better to advance understanding and change in this area. And then with further reflection and review of the literature, you can then determine how you would approach answering the research question you had in mind. How has that research question been informed and revised by some of this individual work? And then continuing to walk through the process as you think about the design of the study, what data or information do you need? Do you have the skills to do it all yourself? Most of us rarely do. So this is where you need to be thinking about what skills are needed for the study. Who do I need to collaborate with? Or if there's a study going on that you have interest in, what are you bringing to that group? And then finally, and as said before, if you're thinking about research study, not only what people in human power do you need, but also do you need funding to proceed with the study or not? So hopefully returning to these questions, the big question, but you thinking through sort of your own positionality and what you care about, what you might do in the near or distant future, hopefully that gets you thinking a bit like a researcher, which hopefully sets us up for further conversation about genetics and precision medicine and where there may be some intersectionality. Next slide. So here are some of my key references, and I certainly appreciate your attention and look forward to the subsequent presentations, which will take much deeper dives on the primary topics for today's presentation. And I also invite questions that I can answer at the end. Thank you.
Thank you so much, Nick, for that wonderful presentation on research. I know that all of those on the call today have a lot of intellectual curiosity, and you're providing a tour or providing the building blocks and the knowledge, laying out the knowledge that they'll need to carry out a research project will be will be really supported by the next two topics. So I would like to thank you again and let the listeners know, the participants, that we're going to take a 3-minute break. When we come back from the 3-minute break, fasten your seatbelts for the rest of our whirlwind tour today through genomics and genetics and precision medicine. We'll be back at 48 past the hour. See you shortly. Welcome back, everybody. I hope you had a moment to stand up and stretch your legs. We will continue with the webinar, and it gives me great pleasure to introduce Dr. Nguyen Park. Dr. Park has been a PA since 2003. She's worked in various fields, including genetics, cardiology, nephrology, and environmental medicine. Dr. Park was the first PA chosen for the National Institute of Health American College of Medical Genetics and Genomics Genomic Medicine Program Management Fellowship. She is the Society of PAs in Genetics and Genomics president, founder, and co-author of the revised PA Genomic Competencies. Park is an active member of the Inter-Society Coordinating Committee for Practitioner Education in Genomics, serving on the Direct-to-Consumer Genetic Testing Working Group and the Pharmacogenetic Project Group. She also serves as the National Coordinating Center's Genomic Workforce Committee Chair. Park is passionate about genetics, genomics, pharmacogenomics, and changing how medicine is taught and practiced. I know all of you on the webinar today will be fascinated by her presentation, and Dr. Park, the stage is yours. Welcome.
Thank you, Dr. Multak, for that kind introduction. So today we're going to be talking about genetics in practice, which is my favorite topic to talk about. Next slide, please. I will not be reading the objectives to you, but we'll just do a quick overview. We're just looking at the role of genetics and genomics in clinical practice across the spectrum of our life, looking at some case-based scenarios, which are always fun. We'll look at some red flags that might make you think of a genetic etiology, and then we'll talk about a few common heritable diseases. Next slide, please. So I'm currently working at the NIH, as Dr. Multak said, as an NIH ACMG Genomic Medicine Program Management Fellow, and I've been involved in genetics education since 2011. Some people may not be aware of the NIH Clinical Center, maybe more so since COVID, but I came to the NIH when I participated in the NIH GRI's short course in genetics and genomics for PAs. And I discovered this wonderful public health institution where PAs can work both clinically and in research. For example, previously I worked for the NIH's nephrology consult service. My primary role was clinical, though I was also an associate investigator on one of the nephrology attendings' clinical trials. Other PAs who work at the NIH might have more research-based duties and less clinical time. And what really attracted me to the NIH is that the clinical care requirements are very protocol-driven, and it's just a wonderful place for people to practice what I think is very evidence-based medicine. Next slide, please. Unfortunately, though, the rest of the United States is not the same. Clinical practice today tends to be one-size-fits-all medicine. So unfortunately, people in medicine, not usually PAs, are obtaining incomplete family histories. They're not thinking about possible genetic etiologies on their differential diagnosis. They may be prescribing medications frequently without thinking about pharmacogenomic considerations. And as one of the things that the All of Us research program is trying to address, the diverse backgrounds of our patients is not really well understood and not always factored into how we treat our patients. And unfortunately, for those of us who went into medicine to make a difference, not only does one size not fit all, the efficacy of the drugs that we are prescribing isn't that great. So, I mean, these statistics are a little bit older, but unfortunately, that hasn't changed much. So 1 in 4 oncology drugs are useful for our patients. And so you can see that there's a spectrum. There's nothing out there that's 100% effective. We know that, but it's kind of sad and scary that, you know, the medications that we are prescribing for our patients aren't always effective. Next slide, please. And next slide, please. So, and go forward. So unfortunately, not only are we prescribing medications that don't work, sometimes the drugs that we prescribe cost our patient or cause adverse drug events. And so not only is it not effective, which costs our patients both time and money, they can end up in the ED. Go forward, please. So 60% of adverse drug events which landed people in the ER affected women. 25% of those patients were 85 years and older. In the time frame between just 2007 and 2009, there were almost 170,000 annual ER visits. Next slide, please. So some of you might be thinking, I don't know, is precision medicine really relevant to what I want to do in medicine? I know when I went to PA school over 20 years ago, as much as I loved genetics at the time, I really didn't think that there was a place for PAs to be practicing in genetics. Obviously, I was wrong. And Francis Collins, whom you might have heard of, former director of the National Human Genome Research Project, would agree. He says actually that virtually all human disease has a genetic component. Next slide, please. So let's start with what we know already. We know that genetics and genomics affects the practice of medicine across the entire lifespan. It starts with preconception testing. They're doing pathology testing, as with COVID-19 patients right now. Next slide, please. And a lot of people have this kind of misconception that because each individual disease is uncommon, that they're unlikely to see a patient with it. So it's really not worth investing any time into it. Now, the statistic that's listed here, that there are about 7,000 rare diseases in the United States, that statistic came about in 1984. So I recently attended a lecture less than 2 weeks ago in which they did a machine learning algorithm, and they estimate that actually just looking at Mendelian disorders, that's probably triple the number, so over 21,000. Now, using the statistics of 7,000, that means 1 in 10 Americans have a rare disease if you combine all of the rare diseases together. So that means that all of the people who are attending this meeting, either one of you has a rare disease and knows someone who does. And what's heartbreaking is half of those patients are children. And about a quarter of those diseases are lethal before the age of 8. And the majority of the rare diseases don't have an FDA-approved therapy. However, millions of dollars are being invested into research, such as with All of Us research program, to better understand and discover treatment for these quote-unquote rare diseases. Next slide, please. So what is already being done now? As I said, we're doing pathology testing of malignant tumors. They're looking at infectious diseases, as I said, with COVID-19, but they were doing it before that with Ebola, looking at drug-resistant strains of Staph aureus and Klebsiella, E. coli. They're even doing whole genome and exome sequencing. And in some cases, there have been patients who were misdiagnosed with cerebral palsy. After they had DNA sequencing, not only did it point to a new diagnosis, there was a treatment. So the one patient who'd been misdiagnosed for 30 years was found to have a treatable dopa-responsive dystonia. And in 4% of cases with cystic fibrosis, that's caused by a mutation known as G551D. And now a drug called Avafticor has been developed that is extraordinarily effective at treating this disease in individuals with this particular mutation. Next slide, please. So one of the nation's most effective public health programs is the newborn screening program. And every year, over 4 million newborns are screened, and thousands of infants are rescued from disability and death. Newborn screening begins within 24 to 48 hours of a child's birth. It's just a few drops of blood obtained from a heel stick, and the blood spots are sent to a laboratory that is part of the state or territorial public health department. Next slide, please. So there's a definite role for primary care in genetics and primary care. Primary care practitioners will act as genetic gatekeepers. So other guidelines are going to be specifically looking at families that are above average risk for various conditions like factor V Leiden or colon cancer. And so I want you all to be thinking, how many of you think you might be ordering genetic tests for your patients? How many of you think you might be making referrals for genetic services? So clearly you need to all be thinking genetically when you're looking at your patients and your future patients. Next slide, please. So you might be asking yourself, but how do I start thinking genetically? Luckily for you, in 2016, we revised the PA genomic competencies as a guide for primary care PAs. At the time they were originally written, for PAs who wanted to work in genetics, which I'm all for, of course. But when I came on board, I thought, you know, the majority of PAs are working in primary care. Let's make this applicable to all PAs. Next slide, please. So we revised the competencies, and so there's a framework in place to help you make sure you know what you need to know to take care of your patients. And I know this slide is a little bit busy, but if you become familiar with this framework, it's going to help you care for your patients with genetic diagnoses. And that's fast becoming a reality for most patients as we discover the genetic underpinnings of more and more diseases. Next slide, please. And so, you know, here's an easy example. So you're going to be looking at practice-based learning and improvement. So whatever rotation you go to, maybe you can check in to see, like, who do they refer to for their genetic services? And what kind of coordination can you do to help your patients before that? So that helps us to understand kind of the systems-based aspect of caring for our patients, because it's not only a matter of understanding the clinical aspect, but the reality of the practice of medicine. Next slide, please. Okay, so let's look at Mr. T, who is not the guy from the A-team, which probably all of you are too young to remember. Next slide, please. Okay, so I want you to think about, how would you treat this patient? You've got a 19-year-old Caucasian male with chest pain, and he's seen in the ED. And there's probably a lot of you who want to work in family practice, urgent care, or the ER. So this is a good case to begin with. What might be on your differential? And now you've ordered a few tests. So troponins negative, EKG's negative. What would you do? Well, hopefully you asked a few salient questions about his family history. Move forward, please. Now with this information, what would you do? So let's look at this. So we see that the patient's father had an aortic dissection in his mid-40s, but he's alive and well. And his paternal uncle was deceased secondary to an aortic dissection. Now, how might that change your plan of action? Next slide, please. Well, unfortunately for Mr. T, even though his mother did tell them that there was a history of family history of aortic dissection, that we'd had this history, asked for the echocardiogram, they disregarded that. And 9 days after he experienced chest pain, Tyler collapsed and died. Not something you would think would happen to a 19-year-old. So up to 20%, that's 1 in 5 of individuals diagnosed with a thoracic aortic aneurysm or a type A aortic dissection have an affected first-degree relative. So if you're out there doing family or sports physicals, it's something you need to be thinking about. Next slide, please. And in fact, there are a lot of common patient presentations with possible genetic causes. They may present to your ED or your urgent care with seizures. They have a history of intellectual disability. Maybe they keep coming back in for recurrent infections. If there's a heart murmur that you're unsure of, or if their lab work comes back odd all the time, then maybe you should be thinking about possible genetic causes. Next slide, please. Okay, so I know genetics can be intimidating for some of us to think about. Some of you in our audience may not have been taught about genetics in depth, but we all know this isn't actually how we perform DNA analysis and genetic testing, although counseling, more specifically genetic counseling, is usually involved. Next slide. So we can break genetic testing down into simple categories that range from very specific to very broad scopes. We can do targeted testing if we have a known diagnosis or a familiar variant, and we do that often as like confirmatory testing. If we suspect a diagnosis, we can do more focused or single gene testing. There are many different types of gene panels we can order if we're concerned about a disease type or a particular area. For example, a patient who presents with seizures might find an epilepsy panel useful. More broadly, we can also order cytogenetics, microarrays, or even more broadly, whole exome sequencing, and that looks just at those genes which code proteins, or most broadly at whole genome sequencing. Now, some people are going to say, well, that could be expensive. And yes, definitely the prices for these different types of tests obviously differ. However, the prices for genetic testing continue to drop. And in some states, such as Michigan and California, and shout out to Michigan and to my Wayne State peeps who aren't on the line, Medicaid will cover genome sequencing. Kind of crazy. Next slide. So let's look at case number 2, baby E. Okay, so baby E is a 5-week-old infant, and he's brought into the ED for inconsolable crying. And in the course of discussing the patient's case, the PA says or finds out that the parents immigrated to the country 9 years earlier. On physical exam, baby E demonstrates downward eye deviation while crying, so the astute PA decides to order a CT scan, and large multiple bilateral hypodensities are noted. So what questions should we ask? Next slide, please. So the astute ERPA asked the parents' country of origin, which leads to some questions regarding consanguinity and family history. And in the family history, it comes out that one sibling was predeceased with similar symptoms 9 years previously at age 11 months, secondary to epileptic encephalopathy. So luckily they were at a hospital where they were doing a study on rapid genome sequencing. And they found in the baby's genome a mutation for a severe B vitamin metabolic disorder, thiamine metabolism dysfunction syndrome 2. And soon the baby was quietly drinking vitamin spike formula, and he's now healthy. So it's thought that likely that his sister died of the same thing. And it was reported that this research that this pediatrician had been doing, that gene therapy across 23 studies in the past decade by his and other groups, genome sequencing had led to a genetic diagnosis for 36% of over 1,800 seriously ill children, mostly infants. In 533 patients, which is 29% of the total, the findings led to changes in medical care, which saved some babies' lives. Next slide, please. So infantile encephalopathy is associated with approximately 1,500 genetic diseases, and we have to start by asking the right questions. You need to be thinking about consanguinity in the populations which you serve, as this will increase the risk of rare and recessive disorders. Without prompt treatment, a lot of these conditions have a resemblance to common conditions, and it can lead to inappropriate or delayed treatment. So I would refer you to healthychildren.org and then the CDC milestone tracker, because a lot of people are not asking about whether or not they think that their children are hitting their milestones. Next slide, please. And our last case, Ms. M. So Ms. M is a 38-year-old G2P2 Hispanic cis female medical coder. And she comes to you with concerns about genetic testing for breast cancer. And she wants to know, her sister was diagnosed with breast cancer 5 years ago at age 42, and it was just detected incidentally on a routine clinical breast exam during a well woman visit. She has 2 daughters, and she's really worried. What is the risk of breast cancer for herself and her daughters? And she wants to know, should she be getting direct-to-consumer genetic testing in order to find the disease early, which is what we all want. Next slide, please. So not only do you ascertain that her sister was diagnosed with breast cancer, but you also on further testing find out that her paternal aunt was deceased with ovarian cancer at age 48. And she really wants to know what's her daughter's risk. Next slide, please. So why is it important to know if your patient's cancer is hereditary? Well, definitely it's going to help us to direct therapy. Again, it's going to help us figure out what's the patient's risk. And the interesting thing about genetics, one of the things that I love about it, and Dr. Hudak had touched somewhat on research and how there are a lot of ethical issues, but in genetics especially, you're looking at not just the patient, but the patient's family members, and it definitely has a ripple effect. Next slide, please. So if your patient decides to have direct-to-consumer genetic testing and they say, well, they cover breast cancer. Well, what you need to know is that, you know, in some of the popular genetic direct-to-consumer genetic testing, and direct-to-consumer genetic testing would be some of the big names that, you know, you might see the 50% off special on Black Friday, and, you know, family members are all getting this. But if you, and I know that this is small, but there are only 3 breast cancer variants out of the hundreds out there that could be tested that are actually looked at. And unfortunately, the ethnicities are not fully represented. So it's only relevant if your patient is within the Ashkenazi Jewish population, which clearly our patient is not. And so it's very helpful if you can have a certified genetic counselor who can help interpret the results and then relate them clinically. But direct-to-consumer genetic testing is not a clinical result. They would have to go on to actually have real genetic testing. So it's a waste of money and of time, unfortunately. It can be fun to find out some of the things, but in general, you also have to consider the fact that only 20 to 30% of cancers are hereditary. 70–80% of cancers, we can't explain it from one single source. As we all know, there's many, it's multifactorial. And so because of that, we should never use direct-to-consumer genetic testing as the sole basis of any type of medical decision-making or any decision-making in general. Next slide, please. So red flags. Who warrants a referral to genetic specialists? So there are several factors that raise the possibility of genetic disease in a differential diagnosis. One major factor is the occurrence of a condition on family members that is disclosed when the family history is obtained. The occurrence of the same condition in more than one family member, usually particularly first-degree relatives, if they've had multiple miscarriages, stillbirths, childhood deaths, those are all suggestive of a genetic disease. If there's a family history of common adult conditions, heart disease, cancer, dementia that occur in 2 or more relatives at relatively young ages, that may also suggest a genetic predisposition. Other clinical symptoms that are suggestive of a genetic disease include developmental delay and congenital abnormalities. You'll look for on physical exam dysmorphologies, which would involve either the heart or the face. So if you're noticing a heart murmur, or growth problems could be suggestive of a genetic disorder caused by an inherited mutation, a spontaneous mutation, some kind of teratogen exposure, or other unknown factors. And the things that you would be looking for would be kind of wide set droopy eyes, could be one, flat face, short fingers, tall stature. These might be considered rare or seemingly mild features, but it would be helpful to send them to a genetic specialist to rule in or rule out a genetic disorder. Next slide, please. When it comes to oncology red flags, if you've got patients who are younger than age 50 who have a cancer diagnosis, or if they've had more than one type of cancer diagnosis personally, if they've got multiple family members with cancer diagnosis, or if they're diagnosed with any type of a rare cancer, if you've got any one of those, they should be referred to a genetic specialist. And what we should be doing is updating and reevaluating the family medical history, at least annually, because we know that family history changes all the time. Number one, people remember things and then they come back and they say, oh, well, I was told this, but actually, my father actually died of this instead. So you never know. And then there are obviously specific oncologic risk factors, looking at the specific types of cancers that are all listed here as well. Next slide, please. So the take-home points for you. Usually 2 or more congenital anomalies warrants a genetics referral. If you've got an individual with unexplained intellectual disability or autism spectrum disorder, they should undergo a genetics evaluation. And genetic counselors are key and should be used as a resource. So you should know where your genetic counselors are in your region for patients and providers, and they're a great ally. Next slide, please. So I know all of you are thinking, wow, that's a lot to remember, but I have compiled all these resources for you. So to help you review all of these are organized into easy to find categories. If you're looking for information about a specific condition, you can look in MedGen. You can look at the ACMG Act Sheets and Algorithms, which list guidelines for providers regarding positive newborn screening results, as well as specific scenarios in carrier screening, diagnostic testing, family history, secondary findings, and also, more and more pediatric patients are surviving, so transitioning them to adult healthcare. If you're looking at information about genetic testing, check out the genetic testing registry. If your patient has testing which reveals variants of unknown significance or uncertain significance, you can look those up in ClinVar. And then there's what I call the phone a friend option. So this can help you, these two websites can help you find a genetics clinic through ACMG, and you can find a genetic counselor in your region. Next slide, please. So one of the allies that Dr. Hudak talked about was peers. So what can PAs with more genetic information and education do? Well, we, by our nature, are collegial, right? So we can increase the genetic literacy of other providers and patients. It helps us to increase access to genetic services for diverse populations because we're actually aware of it and thinking about it. It can help us to intervene in a timely fashion for things that are treatable. And for the things that we don't quite have treatments yet, just resolving that diagnostic odyssey and having an answer for why they are having the symptoms that they're having, it can really resolve things for patients and help to prevent any additional invasive and expensive tests. It helps the families to plan for futures. It helps them to understand any possibility of recurrence. And it shows the patients that there is value in medical research, such as with all of us. Next slide, please. So this brings me to my final discussion. So with the pandemic, the discussions occurring on the huddle and social media about burnout are ramping up, and PAs I know began wondering about whether their current jobs were the right fit for them. So there's many opportunities for PAs to get involved with precision medicine. I encourage you to check out our websites for the Society of PAs in Genetics and Genomics. Student membership is very low. We offer free CME all the time. There are a lot of opportunities that are going to be opening up. You could be doing your capstone project, and you could be looking at what could I use All of Us Research Portal for? You could look at that as a possibility. In newborn screening, there's a revolution going on in terms of the number of diseases that they are going to be working on. And so they are going to need more PAs to be working in newborn screening. Currently it's run through each state's public health department, which employs physicians. And there are about 450 therapies in the pipeline. So newborn screening is going to go a huge expansion. In OBGYN, pediatrics, oncology, psych, you need a PA who's knowledgeable about genetic testing and pharmacogenetic results. And you're going to see lots of questions about direct-to-consumer genetic testing and genetic testing in general. And you're going to need to be able to talk to your patients about the testing and about all the ethical, legal, and social issues regarding this. Next slide, please. Just some of my references. And again, if you have any questions, please feel free to put them in the chat. We can answer them. And then also at the end, you can email me as well. So it's been great talking with all of you.
Thanks so much, Dr. Park, for that fascinating presentation. We're so appreciative to learn from and benefit from your passion about genetics and genomics, and to highlight opportunities for students to become involved. We'll look forward to the questions at the end, and I also want to reinforce our appreciation for your role in helping update the genomic competencies, so we can all be on the cutting edge of our practice, of our clinical practice. We'll get to those questions that are in the chat after the next presentation. So I just want to thank you again. And if we could advance to the next slide, I'd be pleased to introduce our next speaker. All right. Well, the next topic is precision medicine. Dr. Collett has a lot to share with you. And if you were wondering how you're going to learn more about precision medicine and AI or artificial intelligence, Dr. Collett will highlight lots of exciting information for you. Dr. Collett is a professor in the Department of Physician Assistant Studies at the University of Kentucky. Her research interest is in health and healthcare disparities, focusing on the clinical integration of genomics and diversity in precision medicine. In addition, she has extensive administrative experience, and she currently serves as the chair of the Senate Council, as well as the University DEI leadership team. Dr. DeShana Collett received her Bachelor of Science, Master of Science, and a PhD from the University of Kentucky, and has more than 20 years of clinical experience. Welcome, Dr. Collett. We're excited to hear your presentation this morning.
Oh, thank you. Thank you for that introduction and welcome everyone. Yes, today we're going to get into a little bit of precision medicine. So our objectives today are there. It's really, we're going to talk about artificial intelligence. So we're going to define AI and precision medicine, as well as how they correlate with other diseases and things. We're going to identify challenges that for the use of AI and precision medicine. So we'll also touch on just some of those ethical considerations you must think about, and we'll also describe some opportunities for educating PA students and PAs about implementing precision medicine in their practice. Next slide. And so what I want to do first is give you just a short summary of kind of how this all began. So it's been about over a quarter of a century since the field of genomics actually took center stage. It all started within the 1990s. There was about a $3 billion or so human genome project that was initiated. It was an international collaborative research project that focuses efforts on mapping the complete human genome and identifying all of the 20 to 25,000 genes that make each of us human beings. So it makes us who we are. The Human Genome Project was supposed to take like 20 or so years, and it actually was completed in about 15 years. So with the completion of that project, it started to lead to quite a bit of revolutionary advances, as you've already seen from our colleagues, what's kind of out there right now. And so these advances in clinical medicine really out of this became the Precision Medicine Initiative that started. And so this began to really evolve around the end of that Human Genome Project around 2015 with President Obama. And he started this $130 million NIH research initiative that was aimed to understand how a person's genetics, environment, and lifestyle can help determine the most efficient approach to preventing and treating diseases. So this truly was a, all about generating scientific research evidence. It really short-term kind of focused on cancer disease with the long-term goal of looking at the overall health, wellness, and really shifting away from that one size fits all. So you're seeing a lot of the things have come to fruition already, such as increasing health electronic records platforms, ensuring more privacy and protection of health data, increasing access to our own health data, which you see with Epic and some of the programs that we have now, and also increase research dollars and grant money to support research. Next. So as we move from this purely sort of evidence-based model of care, which has been done well, and it's well in providing effective clinical therapeutic guidelines for population health. It does not, however, apply well when we think about subpopulations or individual populations individually, like down to the personalized medicine level. So we're now at a point where there is a paradigm shift. And this emphasis has shifted from that one size fits all to one that focuses more on being preemptive and predictive. So this translates into approaching all patients from multiple levels and also involving the patient. So we're no longer really delivering reactive care, meaning after a disease has developed, but we are being proactive, predictive, and personalizing it in order to have a better understanding of what care we should deliver. So this approach really abandons that reactive find it, fix it model and the one size fits all. Next. So what is precision medicine? So precision medicine is predictive, prognostic treatment and care based not only on an individual's patient's genetic makeup, but their outside stressors are epigenomics. So it's tailored to the person, and it is a systems approach to biology and medicine. So we know disease processes differ, right, from one individual to the next. Precision medicine is sometimes framed as a P4 medicine model. So P4 medicine is being, again, proactive and not reactive. What you see in the diagram is that P4 vision of medicine. So it's predictive, preventive, personalized, and participatory. So this results in multiple parameters being used to assess just one individualized patient. So predictive is the model for healthcare delivery. It is patient-centered with coordinated care, which includes like wellness programs, data analytics, genomics, multiomics, data sharing, research. There are specific risk factors that are usually identified here. So we're looking at intrinsic, so such as genetic polymorphisms, even medical history, family history. We're also looking at extrinsic things such as socioeconomic status and personal medical history. Prevention looks at focusing on addressing the risk before any disease actually develops. So tailored prevention or treatment approaches that utilize patient-specific genomics, behavior, lifestyle data, environmental data. It includes intervention and prevention strategies. So think of this such as vaccines come in here, lifestyle intervention, so diet, exercise, tobacco cessation. So the focus is on the individual and not on population. So that personalized medical piece focus on identification of each individual's health risks and early stratification. So it's promoting health behaviors, preventing disease, treating them with precision. And the most important piece I feel out of this as well, that is really kind of new more than the evidence-based medicine piece, is participatory. So patients are taking ownership in their health and will take a more participatory approach in their healthcare with like mobile health devices, which we've talked about earlier, and the availability of individual health data. It's really now instantaneous. We can look on our Apple Watches, we can look at all types of devices that we have that will tell us if our heart rate is up, it will tell you if you're having an arrhythmia, all types of things. So it promotes really shared decision-making processes in the clinical, in a clinical therapeutic approach. So this is really understanding health and disease with precise care. Next. So we know we've said precision medicine really benefits those individuals and those subpopulations of individuals. But with all of these new advances, we still have to think about what are potential barriers. This is not open to everyone, right? The access is there. So we have to consider who will benefit and who will actually be left out. We need to think about the ways in which we ensure that all patients benefit, especially those who are under-resourced and underserved. So you can see here, gaps that we have: provider education, availability of testing, genetic testing, availability of genetic counselors. Reimbursement is a big deal when we look at precision medicine and genomic testing. And we see healthcare disparities that come with that reimbursement. So there's lack of access. Not all health systems have integrated this type of approach within their electronic medical records. So it's easy to understand, easy to approach, easy to see, and to coordinate care. And there's still some lack of legal and regulatory framework around this area. Next. So what you see here, so as mentioned by my colleagues, we know that with all research, we must have a diverse population of subjects for our outcomes to be applicable to all. But this is an area we continue to struggle in. So what you see here in this slide is a study by Popejoy and Fullerton. It was over a 7-year period, and they found that a large number of participants in the research samples used in cataloged genomic-wide association studies were participants primarily of European descent. During the study period from 2009 to 2016, they did have a 5-fold increase in diverse participants, with the majority of that increase seen in participants of Asian descent, so those of South Asian, East Asian, and Southeast Asian descent. However, there had not been a significant gain in those populations that are frequently underrepresented and underserved, such as African American, Latin Americans, Hispanic, Natives, Indigenous people, still represented less than 5%. So by not including these sufficient numbers of underrepresented individuals in clinical trials, the variants observed are less are more likely to be misclassified because they are so rare in populations without Asian or African descent. So this is important because false positives and false negatives in genetic diagnosis are more common in people with non-European ancestry. So this, in fact, as we know, can lead to health disparities, particularly in our treatment outcomes. And just a side note, as a reminder, there's no single gene for skin color. So the gene for skin color occurs in several thousand alleles. So what is interesting also to know is that race, when we see studies that really focus on race is actually defined every 10 years by the Office of Management and Budget. So this is your census. It takes place right before the census. And I want to remind you that race is a socio-political and not a biological construct. It is fluid. So it is important that we continue to increase studies that really include genomic ancestry, an area that a background may be from, a person may be from. And so next. Research has also shown that most studies conducted to identify risks of clinical outcomes actually use self-reported race as part of their standard taxonomy for identification of race as a variable, instead of more accurate approach of really trying to look at or utilizing genetic ancestry markers or ethnicity markers. So the problem with using self-reported race is it may not be accurately reflective of the actual genetic information. Therefore, research studies may be producing or providing an inaccurate picture when reporting differences in treatment outcomes just based on race. So even when we're using secondary data from other sources, such as medical records, the patient is likely not the one actually entering in their demographics, and therefore providers may be making assumption based on what they see in front of them for the patient. And we see this happens a lot in our patient SOAP notes in the history section. Remember, historically, we have group diseases to occur with certain races or ethnicities, for example, sickle cell, and it has been identified as one of the things that we want to look at in only people of color or Black descent, but doing so can lead to missed opportunities to diagnose a disease. So in the figure that you see here, this study found that some African Americans can have up to 99% European ancestry, and self-identified European Americans have substantial admixture from African ancestry. Similar findings have been found in Latin and Hispanic populations that have admixture of Native American and African ancestry. So additionally, what we don't know is most of these studies that do use genetic ancestry is whether the phenotype of race is a confounding variable with differences really related to social determinants of health. So those socioeconomic demographic factors. Ancestral background may have genetic variations that are different, but race and ethnicity, remember, social constructs that influence our lives. So we can't use genomics for the purpose of telling a story about ourselves. I think that's an important thing just to remember where our limitations are here. Next. So when we think about, one more click, thank you. When we think about precision medicine, we should think on a much larger scale of how do we apply the integration of a vast majority of knowledge gained to benefit all patients. Because we lack diversity in genomic studies, we also have missed opportunities for discoveries and lack of a greater understanding of the heterogeneity and effect sizes across ancestral groups. And just, reminder, effect sizes are those estimates of risk, which suggest that this gap will produce translational outcomes that are applied to clinical guidelines and drug therapies that may not be applicable to all. Next. So there are many different areas that can encompass precision medicine, from pharmacogenomics, genetics, genomics, AI, research, microbiome testing. All of these parallel advances have changed the way that we research and analyze large volumes of data. So technology now is allowing us to process data and make informed decisions like we've never been able to do before. These revolutionary advances through precision medicine initiatives, and particularly through genomic testing, have provided a wealth of data that would be impossible for just us as individual human beings to sit down and filter through with accuracy and reliability. And so this is where artificial intelligence comes into play. Next. So we have all of this genetic and genomic information and data, which is great, but there's so much data that, you know, again, it will take an army of us to really comb through and extract. So artificial intelligence is a set of computational technologies that mimic human cognitive intelligence. So there's two things that you will usually see when you think about AI is machine learning and deep learning algorithms. So using deep learning algorithms, it's basically artificial neural networks. And machine learning is algorithms that improve as they are exposed to more and more and more data. All of these are AI. So they gather large volumes of big data and take it from basically a liability to an advantage. AI helps to identify hidden patterns in large data sets and makes informed predictions and improves our understanding. So AI can look through millions of genetic tests and data points, patient outcomes, and actually find those patterns and relationships between such things as tumor genes, cancer, even specific drugs. So while the genes are important and we look at DNA as a blueprint, the real work is carried out, you know, in the cells by the protein. So there's at least about 6 billion proteins and variations of them. So drugs don't target genes, they target proteins. And this is where AI really gets is really helpful for us. AI is being used in drug discovery, development, improving communication between physicians and patients, even transcribing medical outcomes and documents such as prescriptions in remote treatment of patients. Where can you see AI, machine learning, or where do we see it mostly, and what can it do? So right now we're seeing a lot of things. It can identify facial phenotypes of genetic disorders using deep learning, so syndromes that have recognizable facial features. It can diagnose a disease, identify primary cancers from biopsy, predict risk and prognosis of a disease. So think about our continuous glucose monitoring machines and our wearable sensors. It can improve patient and provider communication. So that sort of clinician-oriented, patient-oriented communication. Predict global hotspots using tracing mechanisms. Look at COVID and how we traced it over time. And improve the function and prediction of gene editing tools such as CRISPR. Next. So all of this actually comes with some challenges, right? There's never just a one all done, and this is great. You know, AI has potential to revolutionize precision medicine and by improving disease diagnosis, predicting treatment outcomes, and enabling personalized therapy. However, there are several challenges that need to be addressed to ensure that AI is used effectively and fairly in precision medicine. Some of the key challenges, as we see here, are fairness and bias. So AI algorithms can be biased if they are trained on data that is not representative of the population. For example, if AI algorithm is trained on data for a particular race or gender, it may not perform well when used to diagnose or treat a patient from a different population, which can lead to disparities in accuracy and predictions for different population. If AI models are designed to account for bias in data, they can perpetuate existing disparities in healthcare. So addressing bias and fairness in AI is essential in avoiding exacerbation of existing health disparities. Socioeconomic factors such as income, education, access to healthcare can have a significant impact on health outcomes. So AI may not be able to capture those factors or variables and therefore may not be able to provide that accurate prediction or recommendation for treatment. So it's important to consider that data safety and privacy. It relies on a large amount of data to train and improve its algorithms. However, the data must be kept safe and secure to protect patients' rights and privacies. So there's always a concern about the potential of data breaches or unauthorized access to sensitive health data. So we've seen this already has occurred. We've seen where data has been stolen or some places been hacked. So those are challenges that we have, regulatory challenges, including, you know, questions around liability and accountability, as well as approval of oversight and integration within the clinical workflow. So AI must be integrated in that day-to-day clinical workflow in a way that is efficient and easy to use, or people will not use it. If the integration is not well designed, it's going to lead to clinician burnout, dissatisfaction, which can limit the adoption of any AI related to precision medicine. Next. So where are we really kind of using AI? You'll see a lot of things in the patient-provider care, centered care. So from even 2018 to 2021, which is a small amount of time, the number of AI-enabled medical devices reviewed by the FDA increased by 83%. Most of the tools being found in specialties of radiology or cardiology. What I have here are just kind of different tools for you to look at, but there's, up here at the very top where you kind of see an ultrasound, that's an IDX-DR FDA approved AI diagnostic system that analyzes images of the retina for signs of diabetic retinopathy. So this is actually being used in some primary care settings, and they're using biomarkers for this. This capsule up here is really at the microbiome level. So this is Atmo gas capsule. So this is a gas sensor, profiles the GI system over 72 hours, and it's actually linked to an app. So it uses biomarkers for diagnosis and target treatment of IBS, IBD, malabsorption. There's also something called a GI Genius, and that's actually used to highlight, actually during a colonoscopy, it will actually highlight portions of the colon where there may be potential lesions or polyps or suspected tumors in real time during the colonoscopy. AI also has wearables. We talked about the Fitbit before, but there's electronic tattoos, stick-on sensors that measure respiratory rate, blood sugar, CPM. So it empowers the patient and reflects on that participatory part of precision medicine, and it integrates technologies into that foster that sort of communication between the patient and provider. As I said, the continuous glucose monitoring, and also up above you'll see where we are using it for even medication adherence. So Abilify MySight is an FDA approved FDA approved device where it can actually let the patient know that they've missed their dose, how long ago it's been since their last dose, can remind them to take their Abilify. And there are other things that are used by consumers, such as the Fitbit and the Oura Ring that looks at circadian cycle, overall health benefits, looks at their sleep pattern, tracking meditation, and so forth and so on. So next. We also have clinical applications of AI. Can you click maybe twice? The crisis text line is one of those. So, it's a suicide prevention hotline in which people text instead of calling as a mode of communication. AI analyzed over 100 million texts for clinicians that were sent to clinicians to have a better understanding of what were the patterns in the text that would give them an idea of those individuals who needed immediate emergent care. So what words do you think would most likely appear in a text message for those in danger of immediate self-inflicted harm? Most of us would think probably suicide, die, cut, kill. No. In fact, it was ibuprofen, Advil. Those words were 14 more times likely to be texted and used if they needed emergency care by someone who was going to, as opposed to someone who used the word suicide. A person using the crying emoji was 11 times more likely to need an active rescue. So those who were serious used words such as never, everything, anymore, always. So, now we have AI incorporated into different sort of technology where they can listen to a conversation and recognize depression through the range and the pitch of a patient's voice. MindStrong is another app that has been currently tested out and used, trying to measure brain function and mental cognition and emotional health through consumer smartphone use. And so looking at how you scroll, how you type, the frequency, detection of characters, it has the ability to recognize whether there's an impeding sort of mental crisis that's occurring or that may occur. Next. And so you also see precision medicine in areas such as pharmacogenetics. So, there is only around 1% difference between the DNA letters of any two people. So this is partly why one person's progression of diabetes is different from another person's progression of diabetes. So any drug that takes into account those differences can be called targeted drugs. And pharmacogenetics is the study of how genetic differences influence the metabolic pathways of drugs, or how someone responds to a drug based on their genetic makeup. So we look at all of these things, and there are resources out there that you can use once a person has had that sort of genetic testing. You can plug in information and actually will output, can you go to the next slide? It will provide a sample of what you see here from CPIC resources for CYP2D6 opioid treatment recommendations. So as we know, CYP2D6 is an enzyme that's highly polymorphic and involved in metabolism up to about 25% of drugs that are commonly used in our clinics today. It's a protein. And so this enzyme is entirely responsible for the metabolism of hydrocodone and codeine, et cetera. So what you'll see is when you have all this information using such a resource, you plug this in, you can tell if somebody is a hyper or a poor metabolizer, intermediate, ultra rapid. So is it going to get out of their system faster? If you have a patient who's coming back saying this medication is not lasting as long, or I'm having these side effects, this is important. We've also seen pharmacogenomics and genetics used a lot in psychiatry, particularly around SSRIs and depression medications, where there's someone, it can predictive, where you can tell whether using certain medications will cause adverse side effects for a patient. Next. So where we hit it, there are several things here. So the healthcare ecosystem, what's going on with the healthcare system integration, academia, research and education, and then those other clinical implications. Next. So with healthcare systems integration, they need to include investing in effective healthcare delivery infrastructures. This is not a one-off. The systems that want clinical decision-making support software have to be incorporated using best practices, being proactive again and not reactive. These include scalable approaches to integrate system-wide clinical decision-making support from multiple aspects. It has to be really proactive in screening and risk assessment. And this has happened and is continuing to evolve. We've seen it happen with Epic, and we've also seen it in other larger healthcare systems such as Johns Hopkins and Geisinger Health. They have already really incorporated a lot of this, and it's helped with the workflow and also the needs to what primary care is seeing, what specialties are seeing, but we all are still coordinating care together. So it's that clean decision, clinical decision support. Next. And then the future of how we are educating. So education will be at a more complex molecular biological level as we move on. We're going to really see more incorporation in PA education, medical education, building competencies in analytical tools and catering to learning styles, so integrating online learning, online simulation around this, engaging with computational tools. So learning how to evaluate and analyze data pretty quickly and accurately. And, education will encompass a lot of need to know how to verify, accept, and challenge some of that computational based conclusions that have been published in the literature. This will obviously create more opportunities for students, to really get into this field and for that outcome, positive outcome to be seen within our patient care. Next. And you may notice that sometimes this, within our healthcare education, how are we educating and what does the current landscape look like? It does include some mixed reality. So it combines this virtual and augmented reality in healthcare. So using AI with augmented teaching. If you use augmented teaching, you have a 50% higher retention rate of information. Surgeons are using augmented reality in the operating room. So you're going to see as students and as clinicians more and more of this sort of AI and mixed reality things be right in your face. And so having that understanding early on and just kind of seeking out information will be so helpful. What I will say is opportunity, if you want to follow someone, the medical futurist is it's a website. Also, you can follow him on Twitter, Facebook, but he brings a lot of information, real-time, sort of up-to-date information on a ton of AI advances in healthcare. It's all medical-based. Next. Clinical practice guidelines. As I mentioned before, by Dr. Park, where to find some of these guidelines. She's placed those in her resources as well, but the Society of PAs in Genetics and Genomics can really point you in a great direction, and you can also find those competencies there. The CDC has a ACCE model for evaluating genetic tests, which encompasses looking at validity, analytical, clinical validity, clinical utility, and also those ethical, legal, and social implications that may arise by using tests. Also, the American Society of Human Genetics and AMA has several resources, including, how to talk to your patients about the lack of sensitivity and selectivity of direct-to-consumer testing. Also, the availability of CME when you do this. AAPA also has a ton of modules that you can go to and also get CMA. So lots of learning modules are available to anyone who is a member of AAPA. Next. Other resources, actually go to the next slide, sorry about that. So other resources that you can find, the Medical Genome Initiative is a consortium funded by leading institutions with a significant interest and experience in evaluating human genomes. Also, Vanderbilt has created a website for community oncologists that look at basically precision medicine and oncology. And the Undiagnosed Disease Network is one I will just really kind of point you to. It's a really neat consortium of several different medical institutions across the United States. I think it's funded by the NIH, it's about 12 sites. And they actually find quite a bit of unknown, undiagnosed sort of syndromes that no one could find out what was going on with the patient, and they have found it. So I would go there just to get some information relative to that. And then the next slide is just additional resources that you can look at and find, providing dosing guidelines to those who have undergone genetic testing. Also, just online open research journal publishing and publications that provide evidence-based information. Next. So what is the future? What's next? Patient-centered and focused on the individual is going to be definitely, it currently is and will continue to be more customizable. Continue to focus on better prediction and prevention, identifying causations and risk factors. And patients will not only be a part of shared decision-making, but will be participatory in their overall health and well-being. So really a nurturing thing. And new data is evolving all the time. Some variants are detected by current, what current NGS methods are out there, but periodic reanalysis of a patient's genomic data has to be offered and has to go, you have to go back to it because more and more things are being found and diagnosed. So the next two slides are really just reference slides, and thank you. So I thank you for this time that you've given me to just provide you a little bit of information on precision medicine.
Dr. Collett, thank you so much for that fascinating presentation and giving our listeners, both the PA students, the pre-PA students, and the PA faculty on the call, an opportunity to know what resources are out there. As PA educators, we should be thinking about augmented reality, virtual reality, mixed reality, and encouraging our students to go to the AAPA and all of us and Medical Futurist. And there's a lot of work to be done to be sure that we keep in step with the progress of technology, and as you suggested, make sure that the impact of precision medicine is implemented or integrated into our evidence-based medicine curriculums. So with that, I want to go. We have a couple of minutes for Q&As, and Rachel has asked us if we have any tips for looking at jobs at NIH or other places that combine research and practice. And I'm wondering, Dr. Park, would you be able to address that first?
Certainly, and it's pretty timely because my fellowship ends in July. So I've been doing job searches, so this is perfect. So if you go to USA Jobs, you can look for National Institutes of Health. You can look for the CDC website as well. You can look at the FDA. There are PAs that work in all of those places. You could also look at most academic institutions will allow you to have some research and clinical time. I would suggest also that the VA, which people don't think of, but they have a huge research component. Actually, some of the most cutting edge things that have happened in medicine occurred at the VA. So there's a great research center there. So the other thing is obviously check out the Society of PAs in Genetics and Genomics, because people reach out to us and we post job postings there as well.
That's wonderful. Thank you so much. A lot of opportunities. I'm wondering, Dr. Schully, have you seen PAs who are working in research and clinical practice together in your experience and the roles that you've had?
I think Dr. Park's comments about jobs that are out there were spot on. I would also consider fellowships that allow for both research and practice to happen. Lots of those at NIH and other academic centers as well. So on the NIH training site, there's a lot of different opportunities that are available that people should go and look at.
Wonderful.
And for PA students, they can reach out to any genetic counseling sites or any genetic medical clinics that they might want to do a rotation with, because that might be a great way to kind of get some experience, get their foot in the door, see if this is something that they're really interested in. I think if you have that curiosity that Dr. Hudak talked about, genetics is a great place if you want to, be a detective. You really help your patients, you get to know them in the long term. Genetics is a great place to be.
Dr. Collett, do you have anything to add, or Dr. Hudak, any other personal advice to support those ideas?
I think that was spot on. I would have actually said the same exact thing. I agree with my colleagues on those answers. And, the job piece, I mean, Dr. Nguyen said everything I would have said, and I love the fact that there are more and more fellowships open. So I would definitely look for those opportunities, even within a hospital setting. They may not be a national fellowship, but you may find there are fellowships or residencies within a smaller hospital organization as well.
Thank you.
And I might just add, if that's okay, that I agree with what has been said. I think it points to some of the networking tips that we talked about, but also, as this is an emerging field, this is a place where we need trailblazers, and it's good to be a trailblazer. You may need sponsorship and support from those in your organization or other organizations, but some of these movement into these areas is not going to happen on accident. So I think being purposeful, being passionate, and getting good guidance and support along the way can really show how PAs cannot just participate in this area, but lead.
Thanks, Dr. Hudak. And that's so important. I love the question itself and the fact that those of you who are signed on today are actively thinking about how to combine a profession of clinical work and research. It's important. It's important for us as a profession to have trailblazers and for you to think for a minute about yourself, about being the trailblazer. And we're so excited that you're thinking about that. I want to open up the Q&A box to see if there's any other questions that anybody would like to add. Barbara's asking about the resources for unknown or rare disease research. Is that something we can help her with or guide her to?
The NIH has the Undiagnosed Diseases Network, and there's the Rare Diseases Network as well. Genetic Alliance is another consumer resource that's out there. There's you could just go to the NIH website and NHGRI's website, and All of Us is part of that as well, and you could look there. I'm not sure in the research portal. That might be a place to be looking to see if there's specific research being done like right now. And that's also a place for students if you're looking to think like, where might I land? If you're looking at who's doing the research, you can see where the other curious minds are maybe studying things that you're interested in. Maybe you could look to see if they're interested in hiring a PA.
Yes. And I'd also like to add, if you're a student or a faculty interested in some of the research that you find out, message those content experts on your own. If you don't know about mixed reality or augmented reality or some of the topics that have been talked about here today, send an email and tell that person, that expert, that you're excited to learn more about that, and can you participate in their research, or do they have any guidance, or perhaps they'd even be willing to be a mentor for you. Maybe a short-term mentor, maybe a long-term mentor, taking that bold first step to strengthening your professional network often comes with really great rewards. We're getting close to the top of the hour, and let me just see if there's any last questions here. There's the All of Us link, the web link, the Join All of Us link. And I just want to extend the appreciation from PAEA, AAPA, NIH, the All of Us program, and the PA Foundation. We're so appreciative that you've joined us here today. We know that oftentimes on many projects you work alone. We know that working together can help us go further. And I would just like to share our appreciation to all the panelists today. And from all of us to all of you, thanks so much for being here. Take care now. Bye-bye.